David Weetman and myself are going to try to give you an update about insecticide resistance mechanisms and the associated molecular markers in dengue vectors. So, as said by Vincent, we are facing a re-emergence of arbovirus disease, and of course we have some vaccine for some of them, but with limited efficacy or insufficient distribution. And the two main vectors for these arboviruses are Aedes aegypti and Aedes albopictus. So you know probably that aegypti is tropical, distributed worldwide, and highly anthropophagic. And also Aedes albopictus, the tiger mosquito, originating from Southeast Asia, is now worldwide spread because of because it's very invasive and also can adapt to temperate climate. So in this context, vector control is very important for limiting outbreaks, and these vectors are highly exposed to insecticide, as said before, from vector control activities, but also from the use of insecticide at home, and also maybe also from pesticides that have been used or currently used in agriculture. So if we look at the distribution of insecticide resistance in these two vectors, it's clear that resistance to all insecticide is occurring in Aedes aegypti in different continents. And in some areas, like maybe Isabelle will tell you about that, like in French Guiana, there is very few alternatives because the resistance is so high. Vector control is totally inefficient. And in albopictus, resistance to organophosphates and DDT has been reported in different continents, and recent studies shows that pyrethroid resistance is rising, especially in Southeast Asia, and also it has been reported in Pakistan recently. So also some alternative control tools are being developed. They are not yet usable at a large scale, and there are also limited efforts for developing novel insecticide molecules or formulations against dengue vectors. So managing insecticide resistance will be crucial for controlling these vectors during the next decade. And what do we have for tracking down resistance? Of course, bioassays can allow detecting resistance, and they are of course useful, but they are very laboring because you need a lot of live mosquitoes, and they are also not informative about insecticide resistance mechanisms. Also, biochemical assays are also useful, but because of the limited specificity, sometimes it's difficult to clearly identify the resistance mechanism underlying the phenotype. So in that context, molecular markers, conversely, they are highly specific, and they can allow high throughput screening of resistance, and this can significantly improve resistance management by allowing an early detection of the presence of resistance gene within populations and monitoring the spatio-temporal dynamics of resistance alleles within and among populations. So insecticide resistance can be caused by different mechanisms. The first one is called behavioral resistance and has been well studied in Anopheles. And it consists on the insect avoiding the contact with the insecticide. But for dengue vectors, this resistance mechanism has been poorly studied, and very few is known about that. The second mechanism is called cuticular resistance, and it consists on modification of the insect cuticle, leading to a limited penetration of the insecticide through the insect body. And again, this resistance mechanism has been studied in Anopheles mosquitoes, but very few is known in dengue vectors. And we have no molecular markers for this resistance mechanism. Of course, in dengue vectors, we also have what we call target site mutations, which are mutations in the protein that are targeted by the insecticide, such as the sodium channel and the KDR mutation. And for these resistance mechanisms, we have some good molecular markers to track this mutation in natural population. But last but not least, metabolic resistance. This resistance mechanism is usually based on the biodegradation of the insecticide molecule by specific enzymes, which are mostly called detoxification enzymes. And this, characterizing this resistance mechanism is a big problem because there is a huge diversity of these enzymes in mosquitoes, and characterizing those that are involved in insecticide biodegradation is very difficult. And again, we have limited markers for these resistance mechanisms and also for sequestration. And most of the markers we have are based on RNA, which are very difficult to implement in the field. So, as a conclusion, there is a clear lack of molecular markers because they are not covering all resistance mechanisms and also not covering all arbovirus vectors because most of these markers have been developed or found in Aedes aegypti, and very few are known in Aedes albopictus where resistance is rising.
Okay, thank you, Jean-Philippe. So I'd like to just now talk about the use of some of the mechanisms for which we do have good molecular markers. So thinking now about tracking target site resistance mechanisms. So primarily these have been looking at knockdown resistance mutations in the voltage-gated sodium channel, the target site for pyrethroids and DDT. They're very common in Aedes worldwide. There's 7 or 8 different mutations known, and they've been, from monitoring, they've been seen to increase in frequency quite rapidly. Just the plot on the right there is from Bill Black's group. This was a study throughout Mexico where they were tracking the frequency of the 1016I mutation and found it to be increasing dramatically. But there are also other mutations, for example, in domain 3 of the sodium channel, 1534. And in fact, re-examination of the data, some additional samples by the same group, actually detected that the driver of the change was actually a combination of the 1016 mutation and the 1534. So that what seems to have happened is that the 1534 mutation gives some level of insecticide resistance, pyrethroid resistance, but then when combined with the 1016 mutation, the level multiplies greatly. So there seems to be a sort of multiplicative effect between mutations. So this is also seen in additional mutations. So S989P is one that's found in Asia and Africa, combining with 1016G and 1534C. And electrophysiological studies have shown that by looking at channel insensitivity in modified xenopoocytes, have shown a massively multiplicative effect of these 3 mutations. So whilst in themselves they don't confer a great deal of resistance, when combined they can lead to very, very high levels of resistance. And this has led to sort of suggestions, and this is studies in Myanmar, that if the 3 are combined, they really need monitoring very closely. At present, they primarily seem to be present on different chromosomes, but if recombined, and this seems to have occurred at least at low frequency in Myanmar, then they could potentially explode. I have to correct this. Sorry, this is the text hasn't quite come out right here. So in Aedes albopictus, the only area in which VGSC mutations have been detected is in domain 3. So there's 1534, and there's actually 3 different variants that have been found here, and these have been actually found quite widespread, including in China, USA, and Greece. And actually more recently there's been one at 1532. So there are a number of target site mutations, but these seem, so far, to be less important for resistance. And so KDR mutations are very readily screened. They need to be screened in sort of groups of potentially synergistic ones. But so far in Aedes albopictus, it does seem that probably metabolic mechanisms are more important. And in general, however useful they are, KDR mutations do remain as partial diagnostics. They're not substitutes for bioassays because they're obviously only capturing part of the phenotype in many populations. And this leads on to metabolic resistance, because this is, in many populations, this seems to be a big part of the phenotype. And the problem really is that there's such a massive diversity of enzymes involved. So from the sort of classical superfamilies, the P450s, the GSTs, the esterases, but there's also many, many others that are implicated frequently in transcriptomic studies, including transporters, and in truth, many enzymes that are just kind of ignored as part of the results because we don't really understand their role. There are also the quite complex detoxification pathways, unlike the relatively simple action of the KDR mutations. And again, by contrast to target site, the conservation is seems to be really quite poor across species and even populations to some extent. Also, the exact way in which they work can be complex. So this can be most typically from overproduction of the enzyme levels by upregulation. This could be related to increases in gene copy number, although potentially could also be linked to cis or trans regulation, leading to potentially sort of hidden molecular markers for the regulation. But also potentially the structural variations that are important, which can interact with overproduction. And most so far have been identified, as JP said, as RNA markers, but these are difficult to move forward with. And there's a really urgent need for DNA-based markers of metabolic resistance. And if we look at some of the what we know about metabolic resistance mechanisms so far, for organophosphates, in general, we don't expect target site mutations to be detected, or we expect them to be unlikely in Aedes aegypti because a double mutation is required. Although recently there was a report in India of the common G119S mutation occurring, which merits further investigation. And it does seem that temophos resistance in general is dependent primarily on metabolic mechanisms. Esterases are implicated, and specific ones have been detected. So, in particular, there's two genes, CCE3A and 6A, which have been detected in strains in Thailand. And the overexpression of these genes does seem to be well correlated with temophos resistance. Also, some suggestion perhaps of mutations in the molecules which might also impact resistance. But also P450s have been implicated as well. This is a study of studies from Colombia and also studies from Brazil, which were looking at resistance reversal to temophos. And they suggested the implication of CYP6 P450s. So there is a potential, noting that P450s are often involved with detoxification of pyrethroids for cross-resistance. And also, John Bontas will be saying a little bit more about this afternoon, I believe. In Aedes albopictus, the same esterases do seem to be involved, which is actually really quite helpful for planning marker systems. So, if we're looking at pyrethroids, then there's a repeated identification of the similar families or similar subfamilies of P450s in pyrethroid resistance. So work from multiple different strains from across the world compared to laboratory susceptible colonies has identified, and these are the ones that are underlined or in blue, these are the ones that are repeatedly identified in studies as being linked with pyrethroid resistance. And some of these, the ones with asterisks, have been shown to metabolize P450, have been shown to metabolize pyrethroids in vitro, which is the sort of gold standard functional validation. But whilst there are genes that are repeatedly implicated, it tends to be from a subfamily. So it's sort of one of groups of genes rather than exactly the same gene every time. And this does seem to be a sort of familiar pattern, which might be linked to the massive explosion of the numbers of P450s in each subfamily. And it does raise a question generally about what's the relative importance of P450s versus KDR mutations. And certainly in some strains we see that KDR mutations, when in combination, can give very, very high levels of resistance, whilst using synergists doesn't seem to have a huge effect. And I think that's probably a question that needs answering once we have comparable markers for metabolic and KDR-based resistance. So if we look at Aedes albopictus, then recent studies from Malaysia, again, this is looking at pyrethroid resistance, and this is strongly linked to the overexpression of CYP6 genes and to a lesser extent CYP9 genes, so P450s again. And these have no KDR mutations. Heterologous expression of CYP6P12 in Drosophila conferred a resistance phenotype to deltamethrin. So that's, again, a good form of functional validation, suggesting the gene is indeed having an impact. But signals of selection were rather inconsistent. And that could make DNA-based markers difficult to find unless there are copy number variants associated, which we're going to come on to in a minute. Also, cuticular proteins were implicated in this study. In fact, some of them were orthologs of some that have been implicated in John Bontas's work, actually, previously in Anopheles. But these haven't been exploited further as yet, and certainly is something that could be followed up. So in general, it does suggest a greater importance of metabolic resistance in Aedes albopictus, and certainly some sharing of general subfamilies of P450s that are involved in overexpression with Aedes aegypti. And many P450s genes tend to be implicated, and this is true of both species, much more so than we see in Anopheles, where specific genes are implicated again and again. And it does suggest that it could be groups of P450s that can act interchangeably, and I think a challenge is to identify what are the key ones and the sort of key players that we need to monitor. Okay.
Thank you. Well, as David said, there's a clear need for identifying novel DNA markers of insecticide resistance in dengue vectors. And what I'm going to present now is the output of a recent project that has been conducted in collaboration with CNRS, IRD, Kasetsar University in Thailand, and Institut Pasteur de la Guyane, and the Rhône-Alpine Center for Bioinformatics, and a sequencing company called Elixio. So the rationale of this project was that, okay, KDR mutations are very good markers, but we need DNA markers for metabolic resistance because RNA markers are not very easy to use in field populations to track down metabolic resistance. And the objective of this project was to combine different next-generation sequencing approaches to identify novel DNA markers of pyrethroid resistance in Aedes aegypti. And we focused on deltamethrin resistance because deltamethrin is most used pyrethroid. And this study has been recently published in Genome Research in 2015. So basically, we started by sampling resistant population all across the world, specifically in regions where there is a strong problem with dengue, and so a very high vector control pressure. So we went to South America in French Guiana, and we sampled populations there with, in collaboration with Isabel, and also in Southeast Asia, in Thailand. But we also used some laboratory-selected population originating from French Polynesia that were selected for approximately 10 generations in the laboratory with deltamethrin. Of course, we also use susceptible populations from the same geographical areas in order to avoid bias due to different genetic background between population and strains. So after that, we tested all these populations with deltamethrin using both dose-response bioassays and WHO diagnostic tests. And we found very, very high resistance in French Guiana, and also a very high resistance in Thailand, and a moderate resistance in the laboratory-selected strain. And after that, we decided to segregate from each resistant population to segregate the most resistant individuals from the most susceptible ones by separating those dying at the lethal dose for 25% of the populations and those surviving at the LD 75. And after that, we extract genomic DNAs from pools of 130 mosquitoes from each population and phenotype. And we captured some specific genomic region using the Agilent SureSelect exon capture system. And these regions were covering approximately 763 genes, covering all resistance mechanisms, all putative resistance mechanisms, including, of course, all detoxification enzymes, like P450s, GST, esterases, UGTs, but also some other genes like cuticle protein, transporters, receptors, and also ion channels, including, of course, the sodium channels. And after capturing this genomic region using the SureSelect system, we sequence these regions with a very high dip using Illumina sequencer system. So after sequencing these regions, which represent approximately 70 million sequence, we mapped them on Aedes aegypti genome. And the good news was that we got a very, very high coverage on target regions. You can see all the little sequencing reads here that align very well on the genome. And then we looked at gene copy number variation that were associated with the delta-metrin resistance phenotype by conducting a differential gene coverage analysis between susceptible population, resistant individuals but dying at a low dose of delta-metrin, and those resisting at a very high dose of delta-metrin. And we found approximately 40 gene amplifications that were associated with insecticide resistance. And interestingly, all these gene amplification were affecting, most of them were affecting detoxification enzyme with a major role of P450s, you can see here. So after that, we looked more precisely at these gene amplifications in the genomic context. And we also compared the gene amplification profiles across different populations from different continents. And we first found that we had different, the gene amplification profiles were highly conserved within regions, but sometimes very different between continents. This may be due to the fact that these populations have different genetic backgrounds, and they are also submitted to different selection pressures, both from vector control but also from the environment. We also found that the overexpression of detoxification enzyme in resistant populations is very frequently caused by gene amplifications, as most of the gene that we found amplified in resistant population were previously found overexpressed by transcriptomic studies. And another good news was that in most of these gene clusters that we identified as amplified in the genome of Aedes aegypti, there were a lot of detoxification enzymes that were previously validated as able to metabolize insecticides. Copy number variation apparently are very good markers of metabolic resistance that is associated with an overproduction of detoxification enzyme in Aedes aegypti. And these new markers are very advantageous versus RNA markers because no labeling expression studies are needed to screen them in natural population. DNA storage is far more easy than RNA storage, and it's now possible to genotype individual mosquitoes, which was not possible using RNA approaches. And then we can follow the frequency of resistance allele within and among populations. It's also very easy, just extract DNA and use qPCR. And of course, it now allows detecting both target site and metabolic resistance genes on the same mosquitoes, which is very important to follow the dynamic of resistance within populations. Then, after looking at CNV, we looked at other genomic resistance markers, and we look at the polymorphism of all these target genes, and we found several non-synonymous mutations in detoxification enzyme that were very strongly linked to delta-metrin resistance. And also this needs further validation. This mutation may represent some variant of detoxification enzyme showing higher insecticide detoxification activities in resistant population. And finally, we recently performed a transcriptomic study on the same samples, and we cross-link transcriptomic data obtained from RNA-seq and genomic data obtained from DNA-seq from promoter regions of these genes. And we identified several promoter mutations that were associated with the overexpression of detoxification enzyme in resistant populations. So this means that when sometimes the overexpression of detoxification enzyme will be caused by gene amplification, but not all the time. And when gene amplification is probably not the cause, it is probably possible to identify mutations in promoter region that are associated with the overexpression of these enzymes. So basically, all this work helped to identify novel insecticide resistance markers that in the future will help to develop novel resistance monitoring tools. In conclusion, as said before, insecticide resistance in dengue vectors is increasing worldwide, and managing resistance will be crucial for the next decade in order to sustain vector control activity. And there has been a significant research effort to identify and characterize insecticide resistance mechanisms since the sequencing of the Aedes aegypti genome in 2007, and also the development of several molecular tools such as microarray, and now we have all next generation techniques plus all genome editing techniques that have been developed. So all these tools are very promising for characterizing resistance genes or mutations and identifying novel markers that could be used for resistance management. But improving all this research output and the implementation in the field required a very wide range of expertise and skills. And this is why we think that the WIN network will provide a very good framework for developing such research and improving resistance management. Thank you very much.